I imagine most of us are here because we care a lot about the Strait of Georgia and the Puget Sound. One of the things that's sort of counterintuitive is that we live in a place that's very, very rich in natural resources, but these iconic salmon species, such as Chinook and Coho and steelhead, have been struggling to survive. And so this is an image that you're probably all familiar with at this point. This is showing the decline in the marine survival of Chinook, Coho, and steelhead since the early 1980s. And it's really, the Marine Survival Project is all about trying to figure out what happened during that period. So, like I say, most of you are probably familiar with the fact that these populations have declined, but not everybody is familiar with the fact that most of that decrease in survival is happening at a very, very specific time in their life cycle. So this is an aerial photo of the Fraser River and the Fraser River plume. So most of that mortality seems to be happening in the first month after they leave the river and enter the estuary. And so the question is, why is that mortality rate so high during that critical period? So these are the primary questions. What caused the decline in marine survival, and what is limiting recovery? And that's a mystery that we'd really, really like to solve. And that's the purpose of the Salish Sea Marine Survival Project. So like any mystery, we always start with a list of suspects. And we divide these into kind of three categories. So the first one are environmental conditions. So any changes in sort of ocean conditions or climate or temperature, those types of things are sort of lumped into these environmental conditions. We're also interested in what are called these bottom-up effects. So things that are going to affect the abundance and availability of their food or the quality of their food that would then impact survival. Things such as phytoplankton, zooplankton growth, or larval fish growth. And then of course there's the top-down effects. And so top-down effects are going to be the things that sort of directly impact the survival of these juvenile salmonids, such as disease, competition with hatchery fish, and of course the predators. And so it's those predators that are the focus of my work. So why do we suspect that predators are an issue? So during this period when survival was decreasing, there was an exponential increase in the number of harbor seals in the Strait of Georgia and in Puget Sound. And so that sort of inverse relationship in the populations is the reason why people suspect that seals may be responsible for this decline. But of course, correlation does not mean causation. And so the really important question is, what is that predation rate? And so the classic way of estimating a predation rate is to go out and collect a whole bunch of seal diet samples. And then you look at the bones that are contained in those samples. And we plug all that into this model that basically takes the amount of salmon bones that are in the diet, multiplies it by the amount of mass that they need per day, and then a number of seals, and that will give you a mass of salmon consumed. The problem with the way that this has been done in the past is that most of those bones cannot go down to the species level. So it'll just tell you that salmon was present in this sample. And that's not particularly useful, right? Because these different salmonid species have different life histories and different conservation status. So the majority of my work has been focused on using these high throughput sequencers or next generation DNA sequencers to get at both the proportions of different species of salmon that were consumed and the individual species ID. So these sequencers are incredible. They are able in an afternoon to produce literally millions of these DNA sequences. And so then we can reconstruct using bioinformatic techniques what the animal consumed. So I'm just going to give you one example from a collection that I did in the Fraser River in 2012. So this image here is showing you on the x-axis, this is the month of collection. The bars here are indicating sort of the average overall diet percentage for that population in that month. So these are harbor seals in the Fraser River, and then the numbers on top of the bars indicate the diet percentage. So you can see this sort of ramp up of salmon consumption starting in the spring and then going into fall. But again, this is not terribly useful information, right? It's only useful if we can get the species out of it. So this is now what we're doing with these high throughput DNA sequencers. And it tells a really interesting story. So basically the seals that inhabit the Fraser River are what we call serial salmon specialists. So they start out in the spring and they're eating these spring Chinook, right? And then as soon as the sockeye come in, they're switching, on this upper panel here, they're switching over to sockeye. And then as soon as the sockeye start to taper off, they switch back to Chinook, the fall Chinook, and chum salmon. And then chum is what takes them through the fall. So again, this is what we can now do with this DNA sequencing technology, is we're getting so much more information. So I've been doing, over the last 2 years, really intensive sampling at these 3 estuary sites, so up in Comox, the Fraser River, Cowichan Bay, and I've also been doing some sampling in the Beltane Islets as sort of a non-estuary control site. And I'm working with a graduate student at UBC also, who is a specialist in fisheries modeling. And what he's doing is he's taking the diet data that I'm producing and he's plugging it into this consumption model. So he's incorporating all that information about the population of the seals, how much food they need, and we're turning that into a consumption estimate. So we've done this now preliminarily for the Cowichan River. And so this is an aerial photo of the log booms in Cowichan Bay. And there are, based on our estimates, roughly 240 seals in that estuary. And based on the diet information that we've been able to collect and that consumption model, this is our preliminary estimate. So they're consuming roughly, between 200 and 400 of those adult Chinook in the fall. But more importantly, they're consuming somewhere between 200,000 and 400,000 of those smolts. And that's that critical life stage. So when you can consider the fact that this is 240 seals, and there are 40,000 seals in the Strait of Georgia, roughly 55 if you incorporate the entire Salish Sea, that kind of predation rate is rather startling. So it's going to be a matter of looking at all of the information that we have and trying to generalize the data that we're producing to create an overall consumption rate for the entire Salish Sea. The problem with this is that it's a mathematical model. And it will always be subject to criticism because it is a model that relies on lots of different assumptions. And so this led me to, I had this sort of nagging thing in the back of my mind that I felt like we could do better than just a model. And so I asked this question: is it possible to measure predation directly as opposed to having to sort of infer it through these reconstructions? And that's when I started thinking about the fact that a lot of the ways that fisheries managers are now tracking the marine survival of these juvenile salmon species is with the use of pit tags, or passive integrated transponders. And what pit tags are, they're basically just tiny little radio transmitters. So as they pass through an electromagnetic field, it induces a current inside that transmitter, and it transmits a radio signal with a unique ID in it. And then it's detected by this in-river antenna that you can see in the bottom picture here. So they transmit that unique ID, and you can tell, okay, I tagged this fish here, it made it to this point, so I know it survived to this point. If you get dropout, then it means that, oh, you've got mortality somewhere in between where you tagged it and where you measured it. Well, in addition to these sort of big in-river scanners, they also make these handheld scanners. So this is what your veterinarian uses to scan your dog or your cat. It's the exact same technology. So all this is a loop of wire, some batteries, and a reader board. So my thought was, hey, why don't we just put it on a seal? And so this is kind of a schematic of this. And the concept was, basically, as these seals are slurping down smolts, those individual pit tag IDs are being detected by the thing on his head. And then what happens is that information gets transmitted up to a satellite and down to our computers. So we don't even have to do anything other than sit at our computers and wait for the data to come in. So this will give us not only a predation rate, but if you've got multiple pit tagging studies going simultaneously, the metadata associated with that pit tag ID will tell you exactly which river that came from. And so that's a revolutionary concept. Now it's just a matter of figuring out how to make it happen. So we took our sort of goofy idea to this company called Wildlife Computers down in Redmond, Washington. And by some miracle, they actually took us seriously. And they built us a tag. So this is, it's a great concept, but it's a very different thing to go from concept to reality. And so if we were to simply turn this RFID reader field on around the seal's head, this tag would only last for about a day. And so we have to do what's called duty cycling the tag, which is intermittently turning it on and off in order to conserve battery life. And so the primary consideration is this swallowing time. And the swallowing time is the amount of time that it takes during a swallowing event or an ingestion event of one of these pit-tagged fish that it's in the actual reed field. And the reason that that's important is that if it's a short time, then we need to sample at a really, really high rate. And so that's going to consume that battery pretty quickly. Versus if it takes a while for that pit-tagged fish to pass through the reed field, you can actually slow it down quite a bit. And that prolongs the battery life of the tag that's on the head. The other consideration is the size of the pit tag. So depending upon the size of the fish that's coming out, we can either put a very, very small pit tag in it if it's a small fish, or if it's something like a coho or a steelhead that comes out a little bit larger, you can put a larger pit tag in it. So if it's a small tag, that reed field is shorter. And so it means that the pit tag is going to be in the reed field for only a very short period of time. Versus if it's a larger tag, that reed distance is larger, reed field is larger, and so you can actually prolong the battery life. So we tested this, basically just the feasibility of this approach at the Vancouver Aquarium, and this was our objective. So we just wanted to measure what those swallowing times were for different sizes of pit tags, and then based on that, what the projected battery life would be to see if we can capture a smolt out migration event. And then we also wanted to see the probability of false detection. So a false detection would be if a fish goes by the head of a seal, gets detected, but doesn't actually get ingested. Okay, so this is Hermes. And Hermes is one of the 2 harbor seals at the Vancouver Aquarium. And you'll notice he's got a patch of Velcro on his head. So we trained him to actually take this tag, allow us to place it on him. And this tag was an archiving tag, meaning that all of the data gets stored. It's not actually transmitted. So we needed to train him to let us take that thing back off of him and repeatedly put it on him. So I'm going to pause this just for a moment. So these 3 panels here are all going to be cameras in different corners of the tank. But the lower panel is the important one. So when that arrow is showing that the record is in a 1 state, it means that there's a pit tag in the RFID field. Versus if it's down, it means that it's not in the RFID field. So I'll show you how this works. So we just dumped in a bunch of pit tag coho. So here's a swallowing event. Now we're going to rewind it and show you what that looks like in the data record. So there he ingests it, and that's the swallowing time. So that's that important response variable for us. And a couple more examples here. There's one, and another one. And this is lazy Hermes trying to catch a smolt. Okay, so the other thing that I mentioned was that potential for false positive detections. And we did get a couple of false positives. And here's a good example of one. So you see, the smolt goes right past the RFID field, but it doesn't actually get ingested. But the important thing is that that's a very, very brief event by comparison to an actual ingestion event. And then the last thing that we did was we wanted to get a closer look at what the actual predation events looked like. And I'll get to why in a minute. That's a GoPro. This is Seal Cam. Yeah. And this is completely unprecedented footage, by the way. Okay, so now we're dumping in a bunch of coho. And it's a free-for-all. So pay particular attention to the motion of the head during the capture attempts. So you see that nose kind of juts out every time he tries to capture a fish. And this is a great example here. And the reason we were able to capture that is because the frame rate of this camera was so high. I mean, it's a split second. So these are the results of our study. So starting with kind of the not so great news, the little bitty tags that are kind of being passed around, those 12 millimeter tags, those are the ones that go in Chinook, that go in the fall Chinook. And so the problem is we weren't able to get very good detections with those, and the ones that we did detect, it was in the reed field for such a short period of time that we'd have to go with the full sampling rate. And so that means that it's not yet ready to work with Chinook. But the good news is it works really, really well with coho or steelhead. And so we got 100% detection with those tags that go into the coho, and based on that we get a deployment length of roughly 15 days. So that'll capture your standard outmigration event. So I'm going to sort of now outline how we're going to accomplish this in the long run. So we accomplished this first phase 1 feasibility testing, and the next phase is to do a controlled test of the full system. And so this is what we're going to be doing at the Vancouver Aquarium Marine Mammal Rescue Facility. So we're going to go capture a wild seal. We're going to take it into a captive setting. And the reason we want to do this with the wild seal is we want to confirm that those swallowing times are appropriate for a wild seal. We also want to test the ability of this thing to transmit its data up to a satellite. And then most importantly, the thing that we're going to do is we're going to try to develop an accelerometer trigger. So this sounds crazy, but an accelerometer is a thing in your iPhone that basically helps it go from portrait to landscape mode. It measures acceleration. So if you put an accelerometer in this tag, you can basically say every time that that thing does a head strike, turn on the RFID board, so that it's not actually sampling unless the animal is actively feeding, and that will dramatically prolong our battery life. The other thing that it will do for us is allow us to sample at a very, very high rate so we can potentially capture those little bitty pit tags that go in Chinook. So this is a picture of one of the great big tanks at the Vancouver Aquarium Rescue Facility. And so that's where we're going to be conducting this study, and the Pacific Salmon Foundation just recently generously funded that. And then the last thing is to do a field deployment. And the purpose of the field deployment is to actually implement this thing to get a predation rate out of it. And so what we're proposing to do is to tag 20 seals, possibly in the Cowichan Bay, possibly in another estuary that rears Coho. And then we're going to combine this with a pit tagging process for Coho, or something that can carry a 23 millimeter tag. So that sounds all well and good, but the problem is you got to catch the seals. And catching seals is a pretty tricky thing to do. And so the way that this has been done in the past for seals that haul out on these log boom sites is to deploy a tangle net. And the problem with a tangle net is you tend to only capture the seals that are kind of young and dumb and not very net savvy. So we need a method that's actually going to capture adult seals. So what we're proposing to do is create a capture platform. And the capture platform will look something like this. It'll have a decoy on it. The seals will haul out on it. It looks just like the log booms they love so much, except it'll have a couple of posts on the sides. Then we push a button, and the sides go up on it. So this is something that we're proposing to build as an alternative to the tangle nets, which are not very effective for capturing adult animals. So this is going to be part of our upcoming proposal. So the objectives of that are going to be to determine that coho smolt predation rate, and more importantly, identify where smolt predation occurs. And if we can identify where smolt predation occurs, that can help us evaluate these mitigation options. So the way that we do that is by combining the head tag with a tag that's on the back that is a GPS. It tells us where sort of the latitude and longitude of that particular seal was when it actually ingested that pit tagged fish. And so this is an example from a deployment that I did during my master's degree with these tags. So this is in the Strait of Juan de Fuca. So what you're looking at, each point is 1 minute in 1 month of data from this particular GPS tagged seal. And what you can see, so the haul out is in the background there, where it's kind of, all these lines are originating. And the seal is essentially a commuter. So this area in the forefront here, that's its foraging area. And it's basically doing laps between its haul out area and its favorite foraging area. And they show really, really high site fidelity to those particular foraging areas. And that's about a 20-kilometer distance between the haul out site and the actual foraging area. So the reason why we want to pair these things up is that we can now say with the head tag, this is where that predation event occurred. Or those are the actual points where it occurred. And so we get not only a predation rate, but we're getting that location as well. And we can use that data to then create maps of where the predation is primarily occurring. And so we've got a new student in our lab who specializes in reconstructing the tracks of seals. And so he's going to be producing these maps and showing the habitat use of the seals. And ultimately, what we want to do is integrate this technology into this much, much larger Salish Sea marine survival project. And so this will be a long-term goal. Hopefully what we're going to do is we're going to create a tag that works with all species of salmon, and it can be integrated into this longer-term project. So the question I frequently get is, why are we bothering collecting data? So we've got a method that works, right? You just go out there and you cull the seals. So the problem with this, the problem with seal culls in general, or indiscriminate culling of seals, is that it rarely meets your goals unless you're constantly culling seals. So you basically have to put that seal population into an extremely repressed state and maintain it in that repressed state in order to see any change in their prey. The problem with that approach is that it also has a lot of collateral effects, unpredictable collateral effects, and so it tends to be shied away from these days as a management tool. An alternative to that is the removal of specialist predators, and this is what they're doing down in the Bonneville Dam. So on the Columbia River, basically those dams have created the ideal place for these pinnipeds to be consuming lots and lots of these endangered fish. And so what they're doing now is they're selectively removing those ones that have been identified as specialists. So that's one possibility for us, as opposed to sort of indiscriminate culling. Because the problem with indiscriminate culling is that a lot of those seals don't actually specialize on the thing that we're interested in conserving. It's really only a subset of the population. But again, this is still really only a Band-Aid. I'd say the bigger question is, why are they able to prey on them at such a high rate? And so this is an image from the Cowichan River. And it used to be that before we got to these rivers, they were really, really dispersed, right? So the estuary was a region where you had lots of little channels with eelgrass, and so as those fish are coming out, there's lots of places to hide. And so when we say change the foraging arena, that's the place where those juvenile fish interact with their predators, and that could be seal predators, or that could be birds, or whatever it is. And so if we change that system from its current state, which is a channelized system that basically just blasts these juvenile fish right out into this ideal seal foraging habitat and haul-out habitat where we put these booms, then you might actually have a situation that does a better job of protecting those fish. But whatever we do, it's going to rely on very, very good information about the behavior of these seals and how they use their habitat. And with that, I'd like to say thank you.